my dear students, welcome to STUDYHIGH.Pk and subscribe, keep in contact for more videos and update. Today we are going to start our next lecture of chapter Carbohydrate, lecture number 3, in which we will discuss glycosidic bonds, how they form. Similarly, we will also discuss the glycosides, its different types, configuration, etc. In last lecture, we have discussed about the physiological significance of pentoses and hexoses. We have also discussed the different type of diseases related to them, like galactosemia, its causes, how you can recognize that a definite patient is suffering from this disease or not, and what you will recommend as a diet so that the patient may feel better. So we have also discussed fructose intolerance and different type of concepts in our last lecture. First, I want to tell you the learning outcomes of this lecture. After completion of this lecture, what are the important concepts you will came to know after completion of this lecture? First of all, the most important thing we are going to discuss is the alpha and beta configuration of monosaccharides and glycosidic bonds. These are the two very important concepts. About 90% of students think that we determine whether a monosaccharide is in alpha form or beta form on the basis of projection of hydroxyl group. If it is projected downward, then students say that it is in alpha form. If this is projected above the aromatic carbon, then students think that it is in beta form. But that is a totally wrong concept. As you can see, this is the pictures of Harper's Illustrated Biochemistry, a recommended book for medical students. In first diagram is a structure of alpha D glucuronic acid. It is in alpha form and hydroxyl group on the aromatic carbon is projected downward. Whereas the second structure is of hydrochloric acid. Again in this picture or structure, the hydroxyl group is projected downward, but it is in beta form, whereas glucuronic acid is in alpha form. So the student think that on the basis of projection of hydroxyl group, they determine it is whether alpha form or beta form. But that is a totally wrong concept. We determine it on the basis of hydroxyl group, not only hydroxyl group, but also on the geometrical isomerism of hydroxyl group with another carbon. So how we can determine what is the basic concept behind it? We will discuss it inshallah soon. And second important thing we are going to discuss is the Fischer projection and Haworth projection. What is the difference between these two projections? These two formula are those scientists which give these projection formula techniques are very helpful in determining the different type of isomerism and alpha and beta configuration of monosaccharide as well. Numbering and naming of glycosidic bond. Glycosides, what are the glycosides? How they form? Is different types O, N, C and S types of glycosides. What are the glycodic glycosides? How then it can affect our human heart? What are their different types of roles in our clinical life, etc. And different in roles of glycoside hydrolases and glycoside transferases. These two enzymes are related to glycosidic bonds. Like glycoside hydrolases involve in breakdown of glycosidic bonds, whereas glycoside transferases involve in formation of glycosidic bonds. So these are the two types of enzymes related to glycosidic bonds, whereas we will also solve some different MCQs. So these MCQs will help you in recognizing or understanding this lecture to the better extent. Now we are going to start our lecture. So first thing that we are going to start is the Fischer projection formula, which was given by Emil Fischer. According to this formula, in this formula, Fischer shows the two-dimensional picture of three-dimensional organic compounds. As almost every matter is in three-dimensional form X, L and Z, but Fischer shows those three-dimensional monosaccharides organic compound into two-dimensional structure. Like as you can see, this is a diglyceraldehyde. It is a two-dimensional structure, and most of the monosaccharides in acyclic form you study is of in Fischer projection, and different type of isomerism you also see on the basis of Fischer projection. And this is a three-dimensional structure which is projecting on this screen. I am going to tell you how Fischer determine or organize this formula. This is a three-dimensional compound, and this is a shadow, two-dimensional shadow on the screen. So in this way, it is a projection of an organic compound on two-dimensional formula. In two-dimensional formula, we can determine further different type of isomerism etc. So what are the uses of Fischer formula? Projection formula. Amino acid structure. Different type of amino acids we represent in two-dimensional form by using this Fischer projection formula. Similarly, monosaccharides, D and L isomerism, all these forms are determined. Our Fischer projection formula is helpful in studying the different type of monosaccharides. So you must know what is the Fischer formula because it is going to help you in further studying our biochemistry. The second important projection formula is Haworth projection formula. It is a three-dimensional representation of organic compounds, especially monosaccharides, whereas Fischer formula is a two-dimensional representation, whereas Haworth projection formula is a three-dimensional representation of monosaccharides or different organic compounds. In this formula, we represent the monosaccharides in ring form rather than two-dimensional acyclic form. Like it is the alpha D glucopyranose, it is alpha L glucopyranose. Both are shown by Haworth projection formula. The thicker lines, as you can see, represent that it is close to the observer because as like we are seeing, it is close to us, it is close to me. Like this is an organic compound and this thing that is close to me is represented by thicker lines that is away from me by thin lines. In this way, according to you, if you can see that these thicker lines are close to the observer, it means that carbon number 3, carbon number 2, carbon number 1 is close to the observer, where carbon number 4, carbon number 5 are away from the observer. So you must know about the Haworth projection formula. This will also help you in recognizing different type of isomerism, alpha and beta configuration. Now we are going to start our next topic, very important, alpha and beta configuration of monosaccharides. So what is the basic concept in determining whether a monosaccharide is in alpha form or whether it is in beta form? So first of all, this is the anomeric carbon. We take it as a center. It is a stereo-centered. And then we see that which is the furthest away from this carbon. This is the carbon number 1 and this is the carbon number 6. This is away from this central carbon. So the isomerism, the geometrical isomerism between this hydroxyl group and the carbon that is away from it determine whether a monosaccharide is in alpha form or in beta form. If the two groups, first the hydroxyl group connected to the first carbon or anomeric carbon, which we will take as a stereo center, and the carboxylate or any carbon that is furthest away from this. In glucose, in furanose, mostly the carbon that is connected here, carbon number 6, it is away from the stereo center carbon. So the configuration between this carbon and hydroxyl group, if both are in cis form, then we say it is in beta form. If the carbon that is away from the stereo center carbon and hydroxyl group both are in trans form, then we will say it is in alpha form. Like here, this is a carbon, this is a stereo center, this is the hydroxyl group, it is projected downward, whereas this is the carbon that is away from the stereo center, it is projected upward. Both are in trans form. That's why we can say that this is an alpha configuration of monosaccharide. That's why we call it alpha. Whereas in iodoric acid, this is the hydroxyl group on anomeric carbon, carbon number 1. It is projected downward, whereas this is the carbon number 6 that is furthest away from this anomeric carbon and it is projected downward. In this way, both are in cis form. As both are in cis form, that's why we say it beta form, beta L ioduronic acid. So this is the basic concept on the basis of which we determine whether a monosaccharide is in alpha form or it is in beta form. Let's see. This is the alpha D glucose. Why it is alpha? Because it is aromatic carbon. It is a hydroxyl connected to the aromatic carbon. To determine whether it is in alpha form or beta form, we will see the configuration of hydroxyl and the carbon that is furthest away. So this is the 6 carbon that is furthest away from the aromatic carbon, and this is the hydroxyl group that is connected with aromatic carbon. It is projected downward, whereas this carbon that is furthest away from the aromatic carbon is projected upward. Both are in trans form. That's why we call it alpha D glucose. Similarly, in galactose, this is hydroxyl group projected downward. This is the carbon number 6 that is projected upward. Both are in trans form. That's why we say is alpha D galactose, alpha D mannose. Similarly, this is the D2-deoxy D-ribofuranose, beta form. In this, it is a carbon number 1 in which hydroxyl group is projected upward, whereas its carbon that is furthest away from this carbon 1 is carbon number 5. This is projected upward. So both are projected upward. So both are in cis form. That's why we can say that this is the beta form of monosaccharide. So this is the basic concept. This is the stereochemistry of hydroxyl group connected to carbon 1 and the carbon that is furthest away. Their correlative stereochemistry or geometrical isomerism determines whether a monosaccharide is in alpha form or it is in beta form. Now we will also solve some other MCQs, so you may recognize what I want to say that. So first of all, we have discussed the basic concept. On what basis we specify or define monosaccharide is whether in alpha form or in beta form. Now let's solve an MCQs. Different types of MCQs can be made from this topic. A number of MCQs, but I will only solve one MCQ here. Like in the given compound, what is the name and configuration of the compound? First, glucose. If glucose is carboxylated, it converts in glucuronic acid. And glucuronic acid, D form is glucuronic acid and its epimer ioduronic acid. Like it is a glucose, but here is the carboxylate form and that is projected towards inside of the ring. It is ioduronic acid. I am first to tell you it is ioduronic acid that we discussed above. So first you must know that what is the structure of glucuronic acid, structure of ioduronic acid, different type of monosaccharides, glucose, galactose, fructose, etc. Now what is the configuration of this? Whether it is in alpha form or beta form, L form or D form. In most of the compounds, the L form is that is, if you can see in above here alpha D glucuronic acid, the COO negative is projected above the ring. That's why we say it is D form. In L ioduronic acid, it is downward. That's why we say L form. It is a Fischer structure which determines whether it is in D form, L form. Here, as you can see, it is alpha D glucopyranose. Why it is alpha D glucopyranose? First of all, why alpha? Because this hydroxyl group is projected downward. This is projected upward. Carbon number 6. Here is carbon number 6. So it is projected upward. Both are in trans form. That's why we say it is alpha. Second, it is D. Because this is C. This 6 carbon is projected from that side. That shows that in Fischer projection it is on right side. Whereas in L form, alpha L, this is projected upward, hydroxyl group on carbon 1. Whereas the carbon number 6 is projected downward. This is again alpha form because both are in trans form. But it is L form. Why L form? Because this 6 carbon shows that in Fischer projection it is on left side. And that's why when the Fischer projection converted into ring form, it is projected inside of the ring. So in this way you can determine whether a ring is in alpha form or beta form. So first of all, in this way, first you must know that this is the alpha for L form of iodinic acid, as this carboxylate group is projected. First of all, you must know that this is L form of iodinic acid because the COO negative, the carboxylate form is in L form in Fischer projection. So you must know in ring whether it is in L form or D form. Second, we are going to discuss whether it is alpha form of iodinic acid or beta form. Now first, it is the anomeric carbon and hydroxyl group is projected downward, whereas this is the carbon that is furthest away from the anomeric carbon and it is also projected downward. So both are in cis form. That's why we can say that it is the beta L iodinic acid. B is our correct option in question number 1. So as like on the basis of alpha beta configuration, different type of MCQs can be made from this topic. Now we are going to discuss some next topic cover glycosidic bond and glycosides and its types. So first of all, I want to tell you what is glycosidic bond. Like when two monosaccharides combine with each other, then they combine with the bond. Like when two glycoside glucose molecules or glucose galactose combine through a bond, and that bond is known as glycosidic bond. So the bond in which a glycosidic oxygen links the glycoside with the aglycone or sugar called O-glycosidic bond. There are different types of glycosidic bond. O-glycosidic bond, N-glycosidic bond, S-glycosidic bond, and C-glycosidic bond. So if the two compounds, one is a glucose and another may be a glycone or aglycone, there are some terminologies which I want to explain before discussing the glycosides and its types. One is aglycone. Glycone means sugar and aglycone means anything other than sugar. So the bond, glycosidic bond, can be present between the sugar and another sugar, or one is sugar and other may not be sugar, like aglycone. Like if this is a monosaccharide that is a sugar, it combines through a glycosidic bond with an other thing that is a steroid or that is alcohol, anything. So aglycone means anything other than sugar. You can say that it may be alcohol like ROH or any steroid molecule, menthol, etc. So first you must know what is the difference between aglycone and glycone. Glycone means sugar, aglycone means anything other than sugar. So the bond in which glycosidic oxygen links the glycoside with the aglycone or sugar called O-glycosidic bond. Like in this is the structure of a disaccharide in which two monosaccharides are connected by oxygen atom. So that's why we can say that it is O-glycosidic bond. If this oxygen is replaced by something other than, like if this oxygen is replaced by nitrogen, nitrogen connects the two monosaccharide, or one is monosaccharide and other is anything other than sugar, then we say N-glycosidic bond. And if this oxygen is replaced by S, it is called S-glycosidic bond. And the compounds that contain S-glycosidic bond are also called thioglycosaccharides, whereas those compounds that contain N-glycosidic bond also called glycosylamine. And if this oxygen is replaced by carbon, then we will say it C-glycosidic bond. So what is the glycoside? Glycosides are the compounds formed by the condensation of a monosaccharide with another sugar or a glycone. Now we are discussing this topic in detail. What is the different types of glycosidic bond? Here I want to show you the different types. Like it is a monosaccharide, sugar. It is connected with this thing by a sulfur atom. That's why we can S atom. That's why we can say that S-glycosidic bond is present between these two compounds. Whereas if this S is replaced by nitrogen, like in ATP, in nucleotides it happens. This is a phosphate bonds This is the ribose sugar. It is connected with adenine through the nitrogen. This is N-glycosidic bond. You must know, they may ask in MCQs or in SCQs, which bond is present between any compound, like which of the following bond is present in ATP. N-glycosidic bond, A-glycosidic bond, O-glycosidic bond. So first of all, both N and O are present in, because ribose connected with this adenine through N-glycosidic bond and ribose connected with phosphate through O-glycosidic bond. So both are present. You must know that. But if they ask which type of bond is present between adenine and ribose in ATP, then you can say the bond between the ATP between ribose and the adenine is N-glycosidic bond, whereas between the phosphate and ribose is O-glycosidic bond. So this is another example in which this is the monosaccharide that is connected with the benzene ring through a sulfur atom. This is a thio. This is sulfur means thio. So this is also S-glycosidic bond. So these are the different types that you may deal with in our coming lectures. Now we are going to discuss naming and numbering of glycosidic bonds. Who we can name or define glycosidic bond, whether it is alpha glycosidic bond, whether it is beta glycosidic bond. The naming of glycosidic bond is basically very difficult, very confusing. That's why we have to refer a book, give a reference of from where I am going to discuss, like according to Lippan's, which is also a recommended book of medical students. According to Lippan, we can determine first numbering is easy. Like if this is a one monosaccharide, it is connected with other. The carbons that are participating in forming glycosidic bond are numbered, like it is a carbon 1 of galactose, it is a carbon 4 of glucose, then we can say it is a 1,4 glycosidic bond. So numbering is very easy. But whether it is in alpha form or beta form, so according to Lippan, I am saying according to Lippan because the basic concept is very confusing or difficult, which is not needed for the medical students. So we say it is beta 1,4 glycosidic bond because this is a galactose and it is in beta form. So why galactose in beta form? You can say this is the hydroxyl group that is projected upward. And as I tell you, the process how you can determine whether a monosaccharide is in alpha form or beta form. And this is the carbon that is furthest away from the aromatic carbon. This is also projected upward. Both are in cis form. That's why this is the beta galactose. It is connected with another alpha glucose through a bond 1,4. Carbon number 4 of, here is the carbon 4 of glucose and carbon 1 of galactose. So it is a beta 1,4 glycosidic bond because galactose is in beta configuration and its aromatic carbon is involving in participating glycosidic bond. That's why we say that it is beta 1,4 glycosidic bond. So it is very important. Many MCQs came from this topic. What type of bond is present between trehalose or sucrose, which consists of glucose and fructose, or say you. On hydrolysis of sucrose by sucrase enzyme, which of the following will be produced by it? Alpha glucose, alpha fructose, beta fructose, all can be produced. So now you can understand how different types of MCQs can be made from this topic. Like first you must know that who or which glycosidic bond is present in the fragments of or components of the sucrose. Like one is glucose, and in which form it is present, alpha form or beta form. As this is downward and carbon 6 is projected upward, so it is in alpha form. And which form present in this fructose? It is projected. This is the carbon 1 whose hydroxyl group is projected downward, and carbon away from it is a 6 carbon. It is also projected downward. So it is beta form. So on hydrolysis, it will yield one alpha glucose and one beta fructose. So these are the two components that are forming sucrose. And alpha fructose will not be produced as a result of hydrolysis of sucrose BIOCHEMISTRY enzyme, which is a normal process occurring in our human intestine. So this is the basic concept which will help you in solving different type of queries, MCQs, concept. Now we are going to discuss the last topic of this lecture, the cardiac glycosides. What are the cardiac glycosides? First I want to explain this. The glycosides that have an effect on human heart and contain steroids as their aglycone component are called cardiac glycosides. As I have explained you in glycosidic bond and glycosides consist of a sugar, maybe monosaccharide, any monosaccharide. So consist of a sugar that is connected through O or N or S, any glycosidic bond with another component that may be glycone or aglycone. If another component that is involved in forming glycoside is sugar, then we can say that it is a glycone component of a glycoside. If anything that is other than sugar, then we can say it is a glycone component of glycoside. In cardiac glycosides, the aglycone component is steroid. This is an aglycone steroid nucleus that involved in forming cardiac glycosides through O-glycosidic bond with sugar. This is sugar, this is steroid molecule, and both are connected through O-glycosidic bond and form a glycoside. And as this glycoside has an effect on human heart, this is another structure. This is the sugar form, a sugar component of glycoside. This is the steroid nucleus or steroid molecule. Both are connected with each other through O-glycosidic bond. So in this way, cardiac glycosides are present in different type of compounds. Now I am going to show you another thing. How they affect the human heart. They reduce the human heart rate and increase its output. Inactivate the sodium ATPase pump, especially strophanthus. Digitalis, this is an example, a picture of digitalis which contains digitoxin that can affect or has an impact on human heart rate. They can reduce human heart rate output. Strophanthus, I want to show you the picture of strophanthus as well. This is a strophanthus plants from which we can obtain ovabane. This is an example of strophanthus. This ovabane can inhibit the sodium ATPase pump. As a result, the regulation of sodium and potassium may be affected. So as a result, ovabane reduce the human heart rate, increase its cardiac output, control or make the human heart rate to contract with the rhythmicity, etc. So it may be toxic in different type of concentrations. So it is the basic structure of ovabane. Strophanthus, it is the sugar form connected to the steroid nucleus through O-glycosidic bond. That we have studied during this lecture is alpha beta confusion of monosaccharide glycosidic bond, naming and differentiation, different naming and numbering of glycosidic bond, as well as cardiac glycosides, who then affect the human heart, its examples, etc. So this is our third lecture. In next lecture we will discuss about disaccharide disease, their sources, their different type of diseases related to them, how you can diagnose them, etc. So thanks a lot. Like, subscribe, share. Thank you, Allah Hafiz.